Okay, so what we're going to go ahead and start with is we are going to start with neurotransmission, which it seems like a lot of people will need help with this, of course. All right, so I'm going to go ahead and go through the different steps of neurotransmission. It's basically a step-by-step process that hopefully, if you remember the bathroom, that might help jog your memory on some of the aspects of what needs to happen with it. I'm going to go all the way uh, through reuptake as well, and so it's going to cover kind of a lot to begin. All right, so we start with the neuron. The neuron is. Hold on, let me get a drink before I begin. Sparkling water. All right, so we start with the neuron. The neuron needs to get excited, get stimulated to a point where it's going to cross the threshold. Now, how does it get stimulated? Well, it can get stimulated by in the process of neurotransmission. If once the neurotransmitters cross the synapse and hit the dendrites of the new cell, it can stimulate the cell past the threshold. Additionally, signals from the brain can stimulate the cell as well, making it go past the threshold. Once it crosses the threshold, then what occurs is that we go through a series of steps down the axon of the cell, down the tail, the big long tail of the cell. In each step, all of these things happen. And as soon as it goes through the kind of like domino type process, it starts off the process in the next section of the axon. So it's like a chain reaction where it's like you have the cell and then one and then another and then another and then another and then another until finally the stimulation pushes the neurotransmitters out across the synapse where they then stimulate the next cell. So once the cell has crossed the threshold, then we get to the beginning of neurotransmission. In neurotransmission, you've got sodium ions and potassium ions. And if you want to have help remembering that, think salty bananas, right? When neurotransmission starts, when action potential begins, what you find is that the charge is negative because you've got negative sodium ions inside and positive potassium ions on the outside. Then you have depolarization, where the potassium flushes in and it changes the charge of the cell, and that's depolarization. From there, we have repolarization. The sodium-potassium pump opens up in the nodes of Ranvier. Those are those little holes in the top of the axon that have where the sodium-potassium pump is located. And then the sodium-potassium move in and out of the axon until ultimately we get to repolarization, where the process begins to go back to where it was, where now all of a sudden the sodium ions are beginning to come back in and the potassium is about to be filtered out. Finally, that creates then the stimulation, which starts the next cell. And then you go through the refractory period where the cell has to take a break and rest before it can be stimulated again. So comparing it to the toilet, if you remember our trip to the bathroom when we looked at how a toilet represents the neuron, you have things that are negative inside. That's that negative sodium ions that are inside. And then you flush it down. And when that happens, it can only go down the axon. It can't go back up. The cell has to get a threshold, just like the flushing of the toilet. And then ultimately, once you go through it all, then you go through the refractory period where the cell needs to recharge, just like the toilet needs to refill with water. From there, once you get to it, creates stimulation all the way down the axon to the axon terminal branches. And then the neurotransmitters are released out of the terminal branches, and then they cross the synapse. The synapse is this microscopic gap. It's about 1/10,000th of an inch. And once they cross it, they find the receptor sites, which are the cell has more than one type of receptor, so it can accept, say, dopamine or serotonin and so on, but it finds the correct receptor sites like a lock and a key, and that stimulates the cell, which causes then neurotransmission in the next part. Leftover neurotransmitters are reabsorbed back into the original sending cell through a process called reuptake. So that is the big long process of neurotransmission, and including reuptake. Is there anybody who have any questions about this? This was a big one and one that a lot of people needed help with. Okay, sounds good. The next one that people needed help with was the older and central brain structures. This is basically a quick run through of those older structures, meaning older evolutionarily. So it's ones that evolved in those early species first before you get to the more advanced areas of the cerebral cortex. So you start off with the brainstem. The brainstem has a couple parts in it. You've got the medulla, which is in the lowest part. It's right near by the neck, or by the neck up here. And then it's what connects the spinal cord to the brain itself. The medulla controls heartbeat and breathing. Then you also have the pons. The pons kind of wraps around the brainstem, or the pons is right above it, I'm sorry. And the pons controls basic movement, usually facial muscle movement. So think like species needed to learn to eat. Then you've got the reticular formation, or sometimes called the reticular activation system. And what that does is it's wrapped around the brainstem itself, and it controls when we are alert and when we go to sleep. From there, what we find is, sorry, my video went out for some reason. I'm not really sure why. Okay, we'll pause it right here. Hold on. So then we have the pons, and the pons deals with basic movement in the face, probably movement for eating and things like that. And then the reticular formation is the other part of the brainstem, and that controls when we wake up and when we go to sleep. Above the brainstem, we have the cerebellum, which is kind of in the bottom area of the brain. It's about the size of a fist. It controls coordination and balance. So it's a pretty big part because it also controls and stores the memories of all our skills that involve coordination and balance. So all of our muscle memory and stuff is recorded there. From the cerebellum, we can go into the limbic system. The limbic system is the three-part system, being the hippocampus, the hypothalamus, and the amygdala. Amygdala controlling negative emotions and the strong emotional response. The hippocampus controlling memories and factual memories especially. And the hypothalamus, which controls the endocrine system, keeps our body in homeostasis, is the main pleasure area of our brain, positive emotions, and so on. Those are the main ones. You also have the thalamus. The thalamus is like the train station of the brain. That's where messages come from all the different parts of the brain. They collect in the thalamus, and then they get sent off to the rest of the body, or the body sends messages back up to the thalamus, and then that sends it to the proper spots in the brain. So you also have the corpus callosum, which connects the two lobes of the brain together. And when it's severed, that's called a hemispherectomy. Those are all the old brain structures that I was going to cover before I was going to switch gears and go into sensation and perception for a while. Those were the two that were asked about the most. Is there any that's really pressing that people maybe need before I move on? Okay, I will take that as a no. So now we're going to get into sensation and perception. From there were a bunch that of the 8 responses I had, 4 or 5 people wanted to hear about. So I'll go through those kind of as we begin here. And two that really go together that a lot of people asked about were absolute thresholds and difference thresholds, along with signal detection theory and Weber's law, which kind of goes with those two. So, when we're talking about absolute thresholds are the minimum amount of information that's needed for your sensory organs, like your eyes or your ears or your nose, for it to get it stimulated so it can register, so that it can kind of that we can see something or experience something, so that our brain can then get an electrical signal that tells us, yes, we're seeing something or hearing something. It's the minimum amount. So like a really, really, really tiny sound or a light that's really, really far away would be our absolute threshold. But a even softer sound or a light that's further away, it's not going to stimulate our eyes or our ears enough. So that's our absolute thresholds. But our absolute thresholds also can change too. Things like motivation and attention can affect how we hear, how we see things. If we're more motivated to see or hear something, we may hear it. If we are more distracted, we may not. So our level of attention plays a really big role with that as well. Additionally, then you've got your difference thresholds. Your difference thresholds are the amount of information needed to notice a difference. So, like if you're in a warm house and then you step outside, your skin is going to feel the change in the air temperature. And that means that it's crossing your difference thresholds. It feels colder when it's windy or rainy. Or later in the summertime, when you step outside and it's 1,000,000 degrees, it's again, your skin is registering the difference between the temperature inside and the temperature in the air. So that is an example of a difference threshold. But we know that because of Weber's law, we know that difference thresholds are not necessarily a specific amount, it's a specific percentage. So especially like with sound, if you're going from volume 10 to volume 20, that's going to be a huge amount of sound. That's increasing by 100%. But 10 to from 10 to 11, it's not enough. But at the same time, if you go from 2 to 4, you may not notice a difference. Or because that'd be too small anyway. So with Weber's law, it's basically that it's a specific percentage that causes us to notice a change, rather than a specific number. All right, questions about that? Okay. Let's start going through some of the other stuff. Transduction. Transduction is basically where the different sensory organs create electrical signals that the brain understands. So, for example, in the ear, when the hair cells, the cilia that's attached to the basilar membrane, which is inside the cochlea, when those hair cells get stimulated by the movement of the fluid inside the cochlea, it creates an electrical signals in the auditory nerve, which is on the other side of the membrane. So you've got the cilia on one side, and you've got the auditory nerves coming out the other, which then go towards the brain. When you get the movement of those hairs, that's where you get transduction, because that's where the signal has now been converted into something that's electric that the brain can understand. When our rods and cones get stimulated, that information then takes that light energy and it creates electrical impulses that travel from the bipolar cells and the ganglion cells through the optic nerve back up to the brain. So every one of the senses has the ability to transduct, and they have a certain area where that occurs, but it's basically where the sensory organ changes the signal from its raw source into an electrical source that the brain understands. All right, so that's transduction. Again, if you have questions, please speak up in the chat. If you need me to repeat anything, I will, though you could always go back and look at the YouTube video as well. Questions? Nope. Okay. Let's see. Okay. There were a number of people who wanted to know about parts of the ear and the process of hearing, which is probably the biggest of all the different step things for the senses. All right. So sound waves are created by the movement of the air. They travel into the ear. The ear flap is the outer part of the ear, and it's shaped this way in order to recognize the frequencies of sound that deal with human speech. And so it travels into the ear canal, the auditory canal, and it the pressure of the sound wave moves the eardrum. So now the eardrum is going back and forth. That movement of the eardrum causes movement in those three small bones, the ossicle bones, sometimes known as the hammer, the anvil, and the stirrup. And those are moving at the same rate that the eardrum is. The stirrup is attached to the cochlea. And so when those movement of those bones occur, then that creates movement of the fluid inside the cochlea. And that movement of the fluid stimulates these hair cells. So the edge of the cochlea is called the basilar membrane. On one side, the hair cells, I just talked about this. On another side, the auditory nerve. When the cilia get moved or get kind of stimulated by the movement of the fluid, then it creates electrical signals in the auditory nerve. The auditory nerve bundles together kind of like a rope, and then ultimately goes to the to the thalamus, and then to the auditory cortex where we perceive sound. It's not that much, right? Okay, it's a lot. Anyways, any questions about that one? All right. Uh, Bottom up versus top down processing. Okay, so this is kind of the main idea when you're thinking about sensation and perception, how we take information in, and then how we perceive the world. So when there's something new, some sort of new stimulation, we often register it with bottom up processing. That's basically my sensory organs take in the information, and then my brain makes sense of all this new stimulation, and then I perceive that information based on how my brain makes sense of it. However, when we have things that we're used to, we may perceive them with top down processing. The way that top down processing works is that your sensory organs are still taking in the information, but now instead of your brain making sense of this raw new information, it uses experience and expectation to help kind of give it clues so that we can faster figure out like exactly what that thing is. Again, it's basically like, if we recognize something, then our brain can say, "Oh, that's the signals that we get when we recognize that show on TV or that sound that we hear of our favorite song." And so rather than trying to make sense of it and being like, this is a new sound, we recognize the sound and we're able to perceive it a little bit better and also a little bit faster. So that's bottom up versus top down processing. All right. Gestalt psychology. Gestalt psychology is a really old form of psychology that kind of fits into sensation perception. And it's basically how we perceive forms and how we group them together. So the simplest one of these is the figure ground relationship, which is also maybe one of the most confusing. It's basically like that in everything we see, there's a figure and a ground. The ground being the background of what it is that we're seeing, and the figure, the thing that's standing out on that ground itself. So, for example, if you look behind me where my thumb is pointing to this little picture, the picture would be the figure, and the wall behind it would be the ground. But you could also say that in the picture, the people in the picture, this is my daughter and one of her friends, the people in the picture are the figures, and everything else is the background. And you can do this with anything. It's basically like, let's say you looked at a person who is standing still on the ground. How do you know that the person is not part of the ground? Well, because Gestalt psychology tells us that we can perceive the differences between figures and grounds. Additionally, with figures, we also group them together. And so that's a big part of Gestalt Psych, is how we group those figures together. We can group them by similarity, so like similar shapes that look together. We can group them by proximity, so if you have a whole bunch of things all together, we'd probably group them together. Continuity, I'm blanking on some of the other ones. Closure was that one where we see kind of an image that's not actually there, but the other shapes kind of create that image, that closure. All of these are how we perceive forms, and how we group those forms together. Questions about that one? Okay, great. Let's see. Motion perception and the phi phenomenon. This is kind of a pretty simple one. The phi phenomenon gives it a crazy name, but ultimately, motion perception is kind of like those flip books that maybe you made in middle school with sticky notes, where you have the guy in each page of the flip note is the guy going further and further until he falls off the cliff or whatever he does. And that's the way that our brain perceives motion, is that we take in individual images through our eyes, and then our brain causes all those images to kind of like blend together so that we see the motion itself. The phi phenomenon is basically the idea that if you look at really fast moving similar figures, you can kind of turn them on or off and it looks like they're moving. It creates movement. I showed a video. If you want, look it up. It takes literally about 15 seconds. It's a bunch of these like black circles that show up on like a blue or orange screen. They show up one at a time, but they're doing it so fast, it looks like the black circle is going around in a circle. Again, how we perceive motion and movement. All right. From there, we're going to get, there's one more. The vestibular sense. Okay, good. This is a one I wanted to hit. The vestibular sense is connected to our sense of touch, and it's basically our sense of balance. It's controlled by the fluid levels in our inner ear. So fluid that's connected to the cochlea in another structure of the inner ear called the vestibular sac, which is also filled with fluid just like the cochlea. And in fact, fluid from the cochlea sometimes can affect the fluid in the vestibular sac through kind of a connecting tube that we call the semicircular canal. When that vestibular sac is filled with fluid and we're at rest, the fluid is also at rest. Think like a bowl filled with water. But then when we move our body position, the fluid also moves. So if I lean over like this, the fluid is going to start to turn over. This sends a signal to my cerebellum, which then using my motor cortex will adjust my muscles so that I can balance and so that I don't fall over. And that's basically what it's designed for. You may remember that in class, I had you spin in a circle because with the fluid in the vestibular sac, it can still be moving even when we're at rest, which is why sometimes we get that feeling of movement like vertigo or things like that. It's the movement or the fluid levels in the inner ear in that vestibular sac. All right. Perceptual sets. Perceptual sets are basically when we are perceiving anything, when we are experiencing the world, we create a perceptual set. That's how we view and how we perceive the world. And most of the time, how we perceive the world is exactly how it is. But our perceptual sets can be fooled. Two things that can fool our perceptual sets or cause us to have incorrect perceptual sets are expectation. So like if you expect that picture of the Loch Ness Monster is the Loch Ness Monster, then you'd see it that way. But if not, then you just see some logs. Or like a kid that expects that monsters live in their closet will perceive shadows to be those monsters, and so on. The other thing can be context. So context effects can definitely affect our perceptual sets. And what context does is it causes us to compare what we're perceiving to something else that's similar. And if that something else is maybe not exactly how we would normally perceive it, we could potentially perceive things to be wrong. You may remember the picture of the basketball players. It's such a good example, I can't not use it. The picture of the basketball players where you've got the shorter one and the taller one. It looks just from the picture that shorter one is like 5'7", but he's actually 6'8" because the other one is just ridiculously tall. So that's perceptual sets. Wavelengths and intensity of light. With wavelengths and intensity of light, basically there's just kind of two aspects to it. This is the electromagnetic energy that's visible to us. Other non-visible energy types include like gamma rays, ultraviolet light, infrared, and so on. But this is the spectrum that we can see. The overall intensity of the light wavelength will determine its brightness, and the frequency of the wavelength will determine its hue. So let's say you have a red frequency that's got really low brightness. Well, then that's going to be something like pink. But that's basically wavelength, the wavelength of the light, and how then the light stimulates your eyes. All right, I'm just flying through these now. Amplitude and frequency of sound is almost exactly like the frequency and wavelength of light. With sound, instead, you've got the frequency, which is the individual pitch of sound, and then you've got the overall intensity of the sound, which is its loudness. Or that's the amplitude of sound. It's the overall intensity and its loudness, its volume. Okay. The kinesthetic sense. We're almost done with sensation perception. The kinesthetic sense is the feeling of our muscles moving. So like, for example, when I close my fingers together, I have nerves inside my fingers that feel the movement of my bones and muscles. This is obviously really important because if we didn't have feeling inside of us, we wouldn't know when things hurt. We wouldn't know when the movement was actually occurring. Great. Okay. With sound amplitude and frequency, this is kind of exactly the same as what you get with wavelength and intensity of light. Yeah, he certainly is, Jordan. And so with this one, it's basically like the amplitude of the sound is its overall volume, just like the intensity of the light, and the frequency is its pitch, just like the hue of the wavelength of light. So you can compare the two together, and they can really kind of go well with one another. All right. The kinesthetic sense. The kinesthetic sense is the feeling of our muscles and bones moving. If I move my fingers like this, what I'll see or what I'll feel is I'll feel the movement of my muscles and bones. That's obviously very important because I know then that the muscles and my limbs are actually moving because I can feel the movement of those bones and muscles. It's related to a sense of touch because obviously it's the nerve endings inside our skin that feel the movement of those muscles and bones. We're almost done with sensation perception. Process of taste, process of smell will be the last two that we cover in here. With process of taste, we've got gustatory receptors on our tongue. It's basically our taste buds, and they get stimulated by the different bacteria and the different receptors for the different flavors then send signals to our brain, but it wouldn't work at all if we also didn't smell the food. Particles of a food go into our nose and stimulate the olfactory receptors, which then sends a signal to the olfactory bulb, and that goes directly to the brain as well. They work together. 80% of what we taste comes from our sense of smell, but also we can perceive taste differently as well based on if the food looks appetizing. Expectation will affect our perception and will cause us then to say, oh, this tastes better. But certainly smell and taste definitely go together, and most of what we taste is actually because we're also smelling it as well. All right, questions about sensation perception? Anything that I didn't cover that people maybe need me to cover, one that like is just desperate. I know there were a few that I kind of glossed over. I'll wait a minute or two if anyone wants to write. Yeah, transduction is basically when the sensory organs convert the raw information like light energy or sound waves or bacteria into electrical signals that the brain can then understand. So, like for example, when the rods and cones get stimulated in the eyes, when that light energy hits it, it stimulates the cells and creates an electrical signal that then travels from the bipolar and the ganglion cells up to the optic nerve, then to the thalamus, and then to the visual cortex. But it only can happen if that electrical signal is there, and its transduction is the idea of how that raw information is then converted or changed into the electrical signal that we understand. You are welcome. Okay, I'm going to cover a few more things in biopsych. I'll go back to bio. Things there that people said they needed help with. There weren't that many more things, but there were a few, so we'll just kind of get into it. The first is neurotransmitters. There's a whole chart in your book of neurotransmitters that you need to cover, that you should probably know. Dopamine, serotonin, um, epinephrine and norepinephrine. God, I'm blanking. Endorphins. And then you also have GABA and you have glutamate. GABA being the inhibitory neurotransmitter, and glutamate being the excitatory neurotransmitter. And what that means with like GABA and glutamate is that GABA will slow down the stimulation that a cell gets, maybe not letting it get to the threshold for action potential, whereas glutamate will speed up the cell, making it more likely that action potential occurs. Those are the ones you should know. Probably you should know what an undersupply and oversupply are for each of those, but because it's just FRQs, maybe not. In a multiple choice, you would definitely need to know it. But those are the main neurotransmitters that you need to know and cover. All right, the next one is the breakdown of the nervous system. All right, so the nervous system is at the top. This is kind of like a hierarchy. You've got the nervous system. It can be split into two parts. You've got the central nervous system. You've got the peripheral nervous system. In the central nervous system, you can split that into two. You've got the brain and the spinal cord. In the peripheral nervous system, you can split it up with the autonomic and somatic portions of the peripheral nervous system. Autonomic covering involuntary actions like heartbeat, breathing, etc., digestion, and somatic covering voluntary actions, mostly muscle movement. From there, the autonomic nervous system can also be split into two parts, where you have the sympathetic and parasympathetic nervous systems. Sympathetic being when the body is in a state of arousal, where you get things like digestion shutting down, and blood flow going to the muscles and also to the lungs to breathe faster, heart to beat faster, even to the senses so that we can see and hear better. But then other things shut off, like digestion. And then parasympathetic is when we go back to a state of calm. And we're going to always go back to a state of calm because our body has to maintain homeostasis. And it's exhausting once we go back to calm. That's the breakdown of the nervous system. All right, the endocrine system. The endocrine system deals with our hormonal response. Basically, it works that the hypothalamus gets a signal that the body is out of homeostasis in one way or another. And so then the hypothalamus will signal the pituitary gland. It's the master gland inside of our brain right there. And then that will signal all the other glands within our body. Once they do that, they secrete hormones into our bloodstream, which causes changes in our behavior and changes in our mood and things like that. Uh, um, the glands you should probably know: the sex glands, the stomach with ghrelin and leptin. It's more for the biological motives of hunger, but still part of the endocrine system. The pancreas, which deals with blood sugar. You've got the thyroids, which deal with our calcium levels and our metabolism. And then, of course, you've got our adrenal gland, which releases epinephrine and norepinephrine as hormones into our bloodstream, even though our brain will also do that with neurotransmitters, signaling the fight or flight response. So that's the endocrine system. But with the endocrine system, I'm also going to we should also remember that the endocrine system also controls our sleep. The pineal gland is one of these other glands signaled by the pituitary gland, telling us to wake up and go to sleep. This is where we get our circadian rhythm. But people did want to know about sleep cycles, so I'll go through that. With sleep cycles, when we enter into our sleep, we start in stage 1, really light sleeping. You get hypnagogic sensation, the feeling of falling, feeling of floating. From there, you drop into stage 2. Stage 2 is those sleep spindles, bursts of intense brain energy. And then stage 3, you've got the slow delta waves, which is really intense but really slow brain energy. Then we go back up to stage 2, back to stage 1, and then into REM. Once we go into REM, that's where we dream, and our eyelids are the only muscles that are moving. That way, when we experience something in the dream, we don't actually move and hurt ourselves while we're sleeping. I know there are some people who sometimes they wake up when they like kick something or whatever. That's probably a hypnagogic sensation in stage 1. So those are the sleep cycles. What else? Neuroimaging techniques. Okay, so there's a bunch of neuroimaging techniques. You've got CAT scan, which takes a solid picture of structures within the brain, hard tissue structures, and then you've got an MRI, which deals with soft tissue structures. These are different ways using either magnetics or using just kind of X-ray to create that imaging of inside our body, but particularly our brain. An fMRI will also show the blood flow that's going to our soft tissues, basically showing kind of how blood is moving through our brain. You've got a PET scan. A PET scan can be used when you want to identify a specific part of the brain that's being active. What they do is they take a radioactive isotope of iodine and they inject it basically into your bloodstream. It decays faster. It's not harmless. It's harmless completely. And when you do a certain behavior and you then have it scanned, the iodine will show up where in the brain is active. So that's a PET scan. We also have an EET. EET. And that's the brain wave activity, like what you see with sleep. So that they put like a cap on you and it measures the different electrical impulses that your brain is doing. So the different kind of brain waves, and it's the electrical ones. I believe those are the 5 that we covered. So if anyone says I'm missing one, then let me know. I sometimes miss one. I think we're good though. Okay. The cerebral cortex and the sensory cortices. This might be one of the last ones I do because we're getting a little low on time, but we'll cover any others that maybe people need some help with. The cerebral cortex is the outer layer of the brain. This is where our most advanced thinking is done, and it's where we also find our sensory cortices. Sensory cortices include the somatosensory cortex, which is located at the very back of the frontal lobe, and then the motor cortex, which is located, reverse that. The motor cortex is the very back of the frontal lobe, and the somatosensory cortex, which is at the very front of the parietal lobe, located basically at the top of your head. The motor cortex deals with muscle movement. Somatosensory cortex deals with our sense of touch and how we experience touch. Then you have your visual cortex in the occipital lobe. That obviously deals with our sense of vision. And we have our auditory cortex in our temporal lobe that deals with our perception of sound. Those are the sensory cortices. We also have smaller taste and smell cortexes, but they're very, very small. Those are in the temporal lobes, by the way, too. The cerebral cortex is also where we find our most advanced thinking. And so, along with the cortexes, we also have what we call association areas. And those association areas are where we do really advanced thinking, like, for example, in the frontal lobe, our prefrontal cortex deals with judgment, consequence, planning. In the visual cortex, it's things like visual recognition and how we recognize the key details to understand what it is we're seeing. So those are the association areas. Humans have more association areas in our brain than any other species on the planet, which is probably why we're one of the most advanced. So even though other animals like whales, for example, have bigger brains, they have less association areas, meaning that they don't do as much advanced thinking. All right. Was there anything else? Agonist and antagonist drugs, a couple of people said this is basically just how drugs interact with the brain. You've got agonists, which mimic neurotransmitters, and you've got antagonists, which block the production or reuptake of certain neurotransmitters, and then that creates then the different kind of reaction. So those are the two kinds of drugs and how they affect the brain. Is there anything else that people wanted that was on there? Nothing else really had more than one vote. So is there anything else that people were really urgent on from biopsych that they wanted wanted me to cover? All right, I will take that silence as a no. Hopefully, this was helpful for you guys. I apologize for my internet cutting out. I don't know why that happened. It just did. We have one more review session on Thursday from 3 to 4. That's going to cover cognitive psych, personality and intelligence, and research methods. Considering there is an FRQ specifically on research methods, I highly recommend that you come to Thursday's one. Again, I'll record it on YouTube. Definitely, I will be going all the way through experimental design because I really think that that's what one of the FRQs is going to be entirely on. So, Thursday, 3 to 4, we'll cover the last few topics. And that's really about it. I hope that if you've got an AP test like tomorrow or Thursday, you guys do well. Good luck and everything. And anything else from anybody?
Levine, for the